Today I'll be talking about some research I did in the Van Doren lab at Johns Hopkins, and it's on double-sex-independent regulation of the follicular epithelium in Drosophila. So this project is about sexual dimorphism, and for those who don't know, sexual dimorphism is the difference between sexes of the same species, and it's a crucial component of development. So you can see how important it is in a species like peacocks, where the male develops so differently from the female. And in Drosophila, the male develops differently from the female too. And it's determined by the number of X chromosomes in the organism. And so if there are 2 X chromosomes, the fly will develop as a female. And if there's 1, it will develop as a male. And this is different than humans because for us, it's whether or not we have a Y chromosome that determines whether we're male or female. So the presence of a Y chromosome makes a human male. But in Drosophila, the presence of 2 X chromosomes makes a fly female and the default is male. So in the Van Doren lab, we're particularly interested in the development of sexual dimorphism in the gonads, which are the testes and ovaries. And the testes and ovaries contain somatic cells that are responsible for germ cell differentiation, and germ cells, which are the cells that develop into gametes, or in other words, eggs and sperm. Double sex is a transcription factor that regulates somatic sex. We understand the sex determination cascade that produces male and female isoforms of the double sex protein called double sex male and double sex female. And we want to know how these different isoforms control the sex-specific development. We can tell that most sexual differences in somatic cells can be attributed to the sex-specific isoform that's being expressed in the fly of double sex. And in its absence, proper sexual dimorphism is not established, and it causes infertility. So here is a diagram of the somatic and germline sex determination pathway in the fly, which is basically the basis for how you go from having 2 X chromosomes or 1 X chromosome to becoming a male or female fly. So I'll start by explaining the females. So the 2 X chromosomes, that dosage of X turns on sex lethal, and sex lethal is this gene right here. And sex lethal regulates the alternative RNA splicing of transformer, which is this gene right here. And that removes a premature stop codon, and it regulates the alternative splicing of double sex RNA, which eventually leads to the specific female protein isoform of double sex. Then in males, since there's only 1 X chromosome, it's sex lethal is never activated, and then TRA is never activated. So the default TRA2, transformer 2, activates the default double sex, which leads to the male-specific isoform of the double sex protein. So here's the anatomy of a wild-type Drosophila gonad for the male and female. You can see that for females, they look quite different from the males, and it starts with a stem cell niche. And in females, the stem cell niche looks like a stack of cells, and it's called terminal filaments. And then as the germ cells develop, they are supported by somatic cells called follicle cells, which surround the developing oocyte in here in an egg chamber. And eventually, a sheath forms around the follicle cells, which is called the follicular epithelium, which holds them together and nourishes the developing egg chamber and the oocyte inside it. For males, there's a stem cell niche called a hub, which is their equivalent of the terminal filaments. And there's no follicular epithelium that we see in the females, creating this structure around the oocyte. So in double-sex null mutants, as in flies where you knock out all of the double-sex isoforms, this is what the gonads become. For a female, it retains most of the aspects of a female gonad, not the pretty structure, but you can see that there are still follicle cells and there's still a follicular epithelium. Interestingly, sometimes female double-sex null gonads get a hub like this. So you can see there's a little hub up on that one. And then for males, it retains some aspects of testes because there are no follicle cells, or there are follicle cells sometimes, but there's never a follicular epithelium. And it can actually have female stem cell niches. So you can see that here that there are terminal filaments on a male because you've knocked out double sex. So this shows that in the absence of double sex, the somatic gonad forms a stem cell niche that is either female or male, regardless of the chromosomal sex of the fly. So you can see that in double-sex null organisms, it doesn't matter whether they're XX or XY, about the same amount of females form terminal filaments as the amount of males that form terminal filaments. And the same goes for male stem cell niches or even ones that have no niche. You can also tell that double sex does not regulate the formation of the follicular epithelium because when you knock out double sex, females retain a follicular epithelium, but males don't have it. So they retain that aspect of the wild type male gonad even without double sex. Interestingly though, males can still have follicle cells. They're just not surrounded by the epithelium. So, if not double sex, what is causing the follicular epithelium to form in XX but not XY organisms? That's basically the basis of my project. So, for my experiments, I start by knocking out double sex so I can look at what other factors could be causing females to form a follicular epithelium but never males. And so one of the first things I tested was sex-lethal expression in the germline. So, sex-lethal expression in the germline, is not needed for the formation of the follicular epithelium in XX organisms. And we can tell that because when you knock out sex-lethal expression in the germline, you still get a follicular epithelium and you can still get follicle cells. And even though, you know, the gonads are clearly not wild type, they're clearly affected by these changes, they retain these aspects of their anatomy. So despite the fact that sex lethal is knocked out, it retains a lot of the aspects of a regular XX double sex null, which makes us think that sex lethal in the germline is not causing the flies to form a follicular epithelium. And you can see that the percentage of gonads with a follicular epithelium is about the same, whether it's XX double sex null and nothing else, or XX double sex null and sex lethal is knocked out in the germline. So, repressing sex lethal in the germline causes some XY organisms to form a follicular epithelium. And this is something that I didn't really expect. You know, it doesn't really make sense given the way that we think that the sex determination pathway works. As you can see, we did not think that sex lethal had anything to do with the way that males develop in the germline. So it's kind of interesting that some of my experiments, as soon as you knocked out sex lethal in the germline, males started to form a follicular epithelium. This is something that I would like to keep looking into because it's really interesting to me, honestly. So you can see that in the XY double sex null, they never form a follicular epithelium. It's worth noting that I don't have that many trials for this, so maybe eventually if I did 100 trials, I'd start to see some XY double sex null males with a follicular epithelium, but it's pretty impressive that in 20, not one of them had it. And then in the few trials I did do of the XY double sex null, where sex lethal is knocked out in the germline, I can already see that a good portion of them have a follicular epithelium. So not sure what's going on there. It'll be interesting for further experimentation. Okay. Yeah, this is just to talk about, again, we didn't expect this to happen for males. So I tested something else too, to see if not double sex, what is causing the follicular epithelium to form an XX but not XY organisms. And first you knock out double sex, because that's the thing that we're trying to look past. And the next thing I tested was TRA in the soma, also known as transformer. Oh, so it's worth noting that males don't have this form of transformer expressed in the soma, only females. So there's pretty convincing evidence that transformer expression in the soma does not control the formation of the follicular epithelium, because once you knock it out, a good portion of XX flies still have a follicular epithelium. You know, here's one with terminal filaments and it's got a follicular epithelium. Here's one with a hub and it's got a follicular epithelium. And then there's the plain XX double sex null for reference. And it looks pretty similar to these. So not a huge effect by knocking out TRA in the soma. Again, just to reiterate, here's the percentage of gonads with a follicular epithelium. You can see that in XX double sex null, the percentage is pretty similar to the XX double sex null with TRA knocked out in the soma. And then I knocked out TRA in the soma and I looked at what happened to the males. And, you know, unsurprisingly, the phenotypes are pretty much the same as a regular double sex null, because, you know, TRA is not expressed in the soma of males anyway. But you can see here's one with a hub and it's got follicle cells but no follicular epithelium. And then here's one with a bunch of terminal filaments, also no follicle cells, no follicular epithelium. Pretty much the same as a plain double sex null. Okay, and here's the comparison again. You know, in some of these males, there was what appeared to be a follicular epithelium, but it really wasn't convincing, and my amount of trials is pretty small, so I would do further experimentation before I would think anything of that. So that'll be interesting to see in the future whether these possible follicular epitheliums I saw amount to be anything. So in future directions for what I want to do next in my research, I want to test sex lethal expression in the soma, because as you saw before, I only tested it in the germline, as well as transformer expression in the germline, because before I tested it in the soma. And I want to see whether these genes affect the formation of the follicular epithelium. I also want to express female isoforms of components of the sex determination pathway in the male background and see whether this causes a follicular epithelium phenotype. So that would be interesting, because it would basically be the opposite of what I'm doing now, where I'm knocking out female things and seeing if I can take away the female follicular epithelium. I would start putting female things into males and see if I could make it happen. So here are my acknowledgements. Huge thanks to all of the members of the Van Doren Lab for helping me to complete this, especially Abigail Dove, who has been my research mentor and guided me with hands-on instruction through pretty much all of this. It's been hugely helpful. So I'm not sure what time I'm at, but I can answer questions if anyone has any.
Yeah, you've definitely got time for questions if you'd like. So if anyone has questions, feel free to either unmute yourself or you can ask them in the chat.
Oh, for argument's sake, I have a couple more things I looked at, and it's just a comparison of the amount of terminal filaments, so the amount of female stem cell niches in the double-sex nulls compared to the double-sex nulls with sex-lethal RNAi. And you can see that they're relatively the same. Sex-lethal RNAi causes a little bit of higher prevalence of a hub. This isn't necessarily the main focus of my research, but it's some little quirks of it that have happened that I'm not sure whether to make a pattern out of or not. And here's the same for TRA expression in the soma.
Excellent. I think you have a question in the chats if you are interested.
Oh, whoops.
No, you're good. You're good.
Let me find it. Oh, I see. Oh, which method did I use to differentiate between different types of cells in the gonads? So what I did was called antibody staining. And if you see these things on the side, let me find a better picture to give a good example. Okay, this is pretty good. If you see these on the side, these are basically the different antibodies that I used. So antibodies are like a fluorescent tag that becomes attached to certain genes when those genes are expressed. So we know that certain genes are expressed in the stem cell niche, and the NCAD antibody tag would then light up red because that's happening there. And we can say, okay, that's a stem cell niche because these genes are being expressed. I'm not sure if that makes sense.
It does. Thank you.
Okay, yeah. Does this generalize to other animals or is it just flies? Yeah, it actually does generalize to other animals. It's not an exact molecule, but I'm not an exact model, but there are these things called Sertoli cells, I believe, in mice or humans, but there's an analog for follicle cells in humans, which is pretty interesting. And granulosa cells, I'm mixing them up because I don't think about mice and human anatomy very much, but I know that there are analogs. Oh, okay.
Oh, you have
my research mentor, Abby, says you are right, they are in humans.
It's always great to have backup, right?
Mm-hmm.
Okay, cool. I'll give it a couple more beats in case anyone has questions. All right. Otherwise, wonderful job, Lauren. Thank you so much for presenting.